Hello, everyone. I am glad to share with you the article published by our research group on IMETA, Clinical Features and Molecular Landscape of Cuproptosis Signature-Related Molecular Subtype in Gastric Cancer. In this article, we identified 16 gene features associated with copper-induced cell death, and classified them into 4 distinct subtypes. These subtypes exhibited varying clinical profiles, biological pathways, mutation landscapes, and immune landscape characteristics. By developing the copper-induced cell death risk score CSRS system, we observed that gastric cancer patients with higher CSRS scores had shorter survival times, which was associated with the tumor's adhesion status and reduced tumor mutation load. Furthermore, by comparing the tumor genomic landscapes between CSRS-High and CSRS-Low groups, we identified potential therapeutic drugs for gastric cancer patients with higher CSRS scores through drug sensitivity analyses, and validated these predictions through molecular biology experiments. We have reviewed the literature and compiled a list of 16 genes associated with copper-induced cell death. We delved into their role in gastric cancer and conducted an enrichment analysis, revealing that these genes are predominantly clustered in areas related to glyoxylate metabolism, glycine degradation, and copper metabolism. Out of the 408 cancer samples, genetic alterations were observed in 81 samples, with CDKN2A exhibiting the highest mutation frequency. We further assessed the interplay between genes related to copper-induced cell death, noting a significant divergence in expression patterns between tumor samples and normal ones. Based on the transcriptomic features of 16 genes related to copper-induced cell death, 4 distinct copper death phenotype subtypes were identified through unsupervised cluster analysis. Notably, the CSC1 subtype exhibited favorable prognostic associations, while the CSC4 subtype was associated with adverse survival outcomes. These observations underscore the intricate interplay between genetic instability, the tumor microenvironment, and gastric cancer progression, suggesting a correlation with matrix invasion and poor prognosis. Subsequently, we examined the distinct functional signaling, mutation patterns, and immune landscapes in 4 death subtypes of gastric cancer. We utilized principal component analysis (PCA) to develop a risk scoring system for copper-related mortality, aiming to quantify an individual's relative risk of death from gastric cancer and to investigate the correlation between CSRs and clinical practice. This risk score was negatively associated with fatty acid and pyruvate metabolism, as well as the citric acid cycle. Conversely, it was positively correlated with features linked to cancer fibroblasts, tumor-associated macrophages, glycogen and glycosaminoglycan biosynthesis, and cytokine receptors. Subsequently, we compared the tumor genomic landscape between the high CSRs and low CSRs groups. The tumor mutation load in the CSRS-Low group was higher, and 5 mutation features were extracted from the genomic data. In conjunction with the Cancer Cell Encyclopedia CCLE and the genomics of drug sensitivity in cancer GDSC1 databases, potential molecular drugs were identified for gastric cancer patients with high copper mortality risk scores. The difference between CSRS-Low AGS and CSRS-High HGC 27 subtypes was verified by Western blot and drug sensitivity test. Western blot results showed that the protein expression related to copper death process was significantly increased in CSRS-High gastric cancer cells, while the protein expression related to copper protein process was significantly decreased in CSRS-Low subgroup. The results of drug sensitivity test showed that NSC 319726, talazoparib, and fapsigargin had relatively low IC50 in CSRS-High subtype gastric cancer cells. The last part is a simple summary of this article.